So when we are trying to understand the development process, people have given models. So a model, a mosaic model of development, where each cell has some or the other, what we call as the uh, developmental factors that contribute to the specialization of this cell, is what we call as a mosaic. So each cell is having a unique kind of a factor that is governing it. So this principle was given by August Weismann. Later on, experiments were done. Wilhelm Rock's experiment was there on frog embryos. So his work also contributed to the mosaic model. So whatever August Weismann did in 1888, that work was supported by Wilhelm's work. So we'll be looking at August Weismann's work a little bit, what Wilhelm Rock's did, and later on, Hans Dreich, what work he has done and what model he has introduced. So that part we'll look. So we see from 5th century BC onwards, the things have progressed in this manner, and we have come to the 21st century knowledge, and we'll see that rapid progress have ultimately happened in the 21st century. So these are the initial drawings that we can look at. That is the original drawings that were given by Malfigi. So he has drawn very intricate diagrams. So we can see this is exactly the way in which we can observe in chick embryo development how the blood vessels develop. We can see the embryo lying at the center over here, isn't it? And how from the egg yolk the blood vessels are have been laid out, and how it is supplying the necessary elements to these developing embryo. So this is very early embryo development. Whatever structures have been drawn here in the early embryo development is exactly what is observed normally in any chick embryo development. Okay, so this we can see is very early in the development, at as early as the second day of the embryo. Okay, so the preformationist theory is what was put forward by a few of the scientists early in the knowledge gaining process. So this is what was their understanding. So you can see there is tiny individual that has been drawn over here inside a sperm head. Okay, so we can see that there's a little child with its head and sitting bowed is there found inside this. So this tiny person is what we call as animalcule. So this is a figure that has been drawn by Nicholas Hartsocker in 1694, supporting a preformationist theory. But this is not the true way in which actually development proceeds. So this is a preformationist, okay, kind of a thinking, and what was their understanding. Okay, so this is not true. Okay, so this was rejected later on, and what was the dominant theory that is now prevailing is the epigenesis. Epigenesis means gradual development, cell by cell, from single cell to multicellular development is what we call as the epigenesis. Now what is Ernst von Bayer's laws? So the Ernst von Bayer's laws basically start by looking at different time intervals in their development. Okay, so we can see that the embryo development is split into early and late developments. So what happens in early embryo development and what happens in later embryo development. So what we are trying to say in the first principle here is that the general principles, general features, general means which is commonly found in all the organisms, which is which can be found in large group. Okay, so the general principle that is commonly found in most of the large group of animals, most of these organisms occur very early in the embryo development. So this is very important principle that we can generalize and find always occurring in organisms. So whatever we find very early in embryo development is always found in any organism you pick up. Okay, so what is an early embryo development? So we see uh, rapid development, that is cleavage. Okay, so there may be there is cleavage patterns may be different, but we can find that the cleavage would always occur in different organisms. Okay, there may be slight variations of how the cleavage process starts, but from the cleavage process what happens is there is a layer of cells that may be accumulated, multicellular structures start to develop, but it is just plain cells right now. So what we get is the three layers that we find later on, what we call as a gastrulation. So these cells get rearranged and form these layers that we can find later on. Okay, so in the mid embryo development, we can find that this germ layers get formed. So occurs in nearly all the organisms, and that is what we call as a general principles, and it occurs very early in the embryonic development. Okay, and what we call is the specialized features, okay, of organisms that we can find in very few groups, always occurs later in the development. Okay, now what is the specialized features? So the specialized features which may come later on. So if the adult structure is formed, so we may find that in human skin there is hair development. So if you look at the fish, there is scales on the skin. Okay, and then in reptilian you may find scales. In birds there may be feathers, and in other organisms there may be like in mammals, apart from hair you can find claws and nails. So these kinds of structures can be formed, which we can call as a specialized. Now these are found only in restricted few organisms, but it is found in the later embryo development stages. Okay, not very early, and it is restricted to few groups. So this general principle is what has been picked up by Ernst von Bayer's observation and has been generalized. Okay, so that is what we are saying in these first two statements. And again, what we are saying is, suppose if the same program more or less appears similar. Okay, now we know the organisms are having higher capabilities and lesser capabilities. So highly developed and slightly less developed. So if most of the features early in development are similar, so we can think in a such a way that the more advanced form of organisms, do they take all the forms of the lower organisms and then develop into what we know as the higher form, or the most complex form. So suppose we take the humans. So does the human get developed by first forming a worm-like structure, and then leading to an insect-like structure, and then leading to uh, suppose a frog-like structure or a fish-like structure, and then a chick-like structure and a rat-like structure, and then ultimately gets to form a human? So does this chain of events occur? So does the higher form take up all the structures of the lower form and then finally develops into the higher form? Is this the way of development? That when it was observed, it was not found, and it is what they have mentioned over here. Instead of passing through the adult structures of lower form, the higher form departs from more from the lower forms when they are having the highly developed structures getting formed. Okay, so it departs away. So in later parts of the development there is deviations that would occur. So if you want to compare, you will have to compare in the early embryo development, and we find that whenever we study the model organisms, we are looking at early embryonic development, not in later embryonic development. And we are trying to pick up all those principles of early embryonic development, and we are trying to find it out in higher forms. Okay, so that has to be looked at. So this principle is very important. Instead of passing through the adult stages of lower forms, the higher forms depart more and more from them, from the lower forms actually. So therefore, the early embryo development of higher animal is never like a lower animal. That is these four principles that have been enlisted by Ernst von Baer. Okay, so hopefully this is clear. And what Rathke did, that is Heinrich Rathke's work that we have mentioned in the timelines of the historical development in the embryo in the developmental biology. We have mentioned that comparative work was done, so we can look at what kind of a comparison he has made. So if you look at the skull development in different organisms, so we can find here fish, okay, here a reptile, crocodile, and here a humans, and we can find that similar this same color structures are what is common. Okay, so when we go back in early embryonic development, you can find that more or less similarly they may be found, but later on as they specialize, we can find that they restrict themselves into a different form of the structure at a different, at slightly different location. But there is that presence of these structures, but in a different form later on the development. So we can see that this blue part, which is the brain case, okay, where actually the brain resides, isn't it? So in fish, it is restricted to exactly top of the head, above the eye socket, and it is more or less flattened.